Okay, here we go. This is on chapter 12, muscles, and we will talk about predominantly muscle function, and we'll look into some of the physiology, but I will give you a brief overview on muscle structure, reminder of skeletal, cardiac, and smooth muscle, which is section 12.1 in your book, but relies heavily on your anatomy background. And then we'll start talking about skeletal muscle contraction as an example. We'll go through the physiology. We'll end up talking about what a motor unit is, a muscle twitch, and muscle tone. And once we've gone through the entire muscle physiology, which is pretty intense, we'll talk about muscle metabolism. So where does ATP come from in a muscle? And we define muscle fatigue. I will then talk a little bit about the mechanics of body movement. That's section 12.1 again, so part of it will be in different sections. And then I'll talk a little bit about smooth muscle, section 12.3, and cardiac muscle, section 12.4. This is an overview of the three muscle types. Figure 12.1, but you can also look at the table that really summarizes the three types. We've gone through this in tissue. Hopefully this is carried over into this area, and skeletal muscle is in shape. The fibers are cylindricals. The cells, the fibers, same thing, are cylindrical in shape. They're pretty long. They're multinucleated, and they are under voluntary control, and we can see striations, distinct striations. When you take a look at cardiac muscle, it's very similar to skeletal muscle, but the fibers are not quite as parallel ranged because the cells themselves, the muscle, the myocytes, the cardiac myocytes are branched, and the cylindrical shape is not as elongated. They're a little bit shorter. They're not as long as skeletal muscles. Cardiac muscles are also not multinucleated. They only have one nucleus. Each of these branched muscle fibers are connected by intercalated disc, which are really visible when you look at cardiac muscles in the slide. This is also where we find gap junctions between different cardiac cells. They're located within the intercalated disc. Cardiac muscle is under involuntary control, so that means autonomic nervous system, that means parasympathetic and sympathetic nervous system, but these individual fibers also show the striations that we see in the skeletal muscles. So that's also good to remember. Smooth muscle, on the other hand, is quite different. The cells are not elongated. The cells are more shaped like a stretched out football, under involuntary control, like cardiac muscle, so parasympathetic and sympathetic nervous system. We do have the absolute same contractile proteins within all three tissues. They all possess myosin and actin. Those filaments that allow a muscle tissue to, or a muscle as an organ to contract, which is their overall function. All three will contract. Big difference is, of course, where they are located, and as I mentioned, how they're being controlled. Skeletal muscles are distributed throughout your body, connected to skin or bone, such as biceps brachii, biceps femoris, gluteus maximus, latissimus dorsi, tensor fasciae latae, or orbicularis oris. This should be all be flooding back into your memory from anatomy. So hopefully that is the case. Cardiac muscles, of course, only located in the heart, and the heart is a very thick wall, and the majority of that thick wall is the cardiac muscle that we see right here. And then, of course, smooth muscles located in your blood vessels, in the wall of your blood vessels, but also along the GI tract and the walls of your GI tract organs. So that's really a brief overview of the tissue, pointing out here a couple of things that might be of interest as well. In terms of function, cardiac muscle contracts very rapidly compared to the other two, but on the flip side, it does tire out easier. And of course, it has extreme adaptability and it can exert forces ranging from a fraction of an ounce to over 70 pounds. I guess it depends on how strong you are. Cardiac muscle, as I said, is of course only located in the heart, contracts at a steady rate set by the heart's pacemaker. We'll talk more about cardiac muscle and of course the pacemaker when we get to the cardiovascular system, which is fairly soon. So specialty about the cardiac tissue is that it has cells that can generate an action potential and therefore self-excitatory. Smooth muscle, really interesting about smooth muscle is that it does regenerate, contrary to the other two. It regenerates, it grows, it goes through mitosis. And we find it in all those places where food travels through, including, of course, also your blood vessels. Some of the similarities, I said that all muscle cells are called muscle fibers and they're somewhat elongated. Skeletal muscle more elongated than the other. Smooth muscle and cardiac, they're really cylindrical shaped and very long. Cardiac muscles are cylindrical, short, but branched, and smooth muscle more like a stretched out football. We do refer to them as muscle fibers. Their contraction depends on two kinds of myofilaments, actin and myosin. You may have heard them be referred to as thin filament for actin and thick filament for myosin. Some of the terminology that you come across in muscle, in a muscle chapter, just as a reminder, sarcolemma is of course the plasma membrane of a muscle fiber. Sarcoplasm is the cytoplasm of course, a muscle fiber. And some prefixes, myo, mys, and sarco, are all referring to muscle. Table 12.1 is a nice reminder and review of that, so take a look at that, please. Contraction is their overall function. Skeletal muscles allow us to move. Cardiac muscle is, of course, responsible for allowing blood to move through our body by having the heart contract and generate a pressure gradient. Smooth muscle helps maintain blood pressure via the, of course, vessels by being able to squeeze and propel, so vasoconstriction and vasodilation. But muscle also is about generating heat, maintaining posture. So muscle is an important part of our functioning systems. And of course, the nervous system oversees the function of all these muscles. Now, I have some checkpoints here, and you should look them up. Checkpoint 1 describes the connective tissue layers associated with the skeletal muscles. That should be figure 12.3, but you also have a little video posted in your module. And 2, how would severing the tendon attached to a muscle affect the muscle's ability to move a part of a body? Again, give that some thought. I will not answer that in this lecture, but I will address this in our Zoom meeting. Functional characteristics of muscle tissue. Of course, they're excitable, they're contractile, they're extensible, and they're elastic, and they show elasticity. They're excitable because they respond to stimuli, and of course, this comes from your neurons. Their contractility is revealed by the fact that you have the sliding filament model that allows the muscle to shorten forcibly. Extensibility, of course, is revealed by the fact that when one muscle contracts, another muscle is lengthened, and then, of course, is an extensibility, the fact that they can stretch and extend without rupture. Elasticity That's the ability for a muscle to recoil and resume the original resting length. All of these four characteristics do decline as we get older, so they function less and less reliable. Here's a checkpoint. Figure 12.3 and 12.5 will help you with these checkpoints. These are all anatomy-related checkpoints that I want you to review. Checkpoint 4, describe the basic structure of a sarcomere. Checkpoint 5, why do skeletal muscle fibers appear striated when viewed through a light microscope? What's that all about? Checkpoint 6, where would you expect the greatest concentration of calcium ions to be in a resting skeletal muscle fiber? These are questions we will address in the Zoom meeting, so please come prepared. Each muscle is served by one nerve and artery, and usually more than one vein. And when we get to the cardiovascular system, I will talk a little bit about, well, actually we focus more about muscle, about cardiac function and blood pressure and blood flow than we do on naming. Again, this is totally anatomy. You need to bring that back. Take a look at it for review. Each skeletal muscle fiber is supplied with a nerve ending, and that nerve ending is usually an axonal terminal, and within that axonal terminal you have a synaptic knob. And the communication between one neuron synaptic knob and a skeletal muscle fiber is what we refer to as the neuromuscular junction. It's at that neuromuscular junction that the contraction will be elicited by the release of a neurotransmitter known as acetylcholine, ACH, as you all know. Contracting fibers require a continuous supply of oxygen and nutrients, and that happens via arteries. And waste, of course, must also be removed, and that occurs via veins. So nerve supply function to stimulate, arteries to provide nutrients and oxygen, veins to remove waste and carbon dioxide. This is figure 12.7 and is just a rough overview of skeletal muscle contraction. And we have the event at the neuromuscular junction where the motor neuron meets the somatic motor neuron, synaptic knob meets the skeletal muscle fiber, which is referred to as the neuromuscular junction. So there's a stimulus, and this stimulus comes from the nerve ending, and that elicits what we call the excitation-contraction coupling that includes the sliding filament model. And so the initiation of the excitation-contraction coupling ultimately has to do with the neuron stimulating the muscle, generating an action potential. That action potential ultimately causes the release of calcium from inside the muscle. So then we have an increased concentration of calcium that allows the sliding filament model to occur, the contraction to occur, and we either have a full contraction of the organ or it's just simply a muscle twitch. So the excitation-contraction coupling really is the linking of electrical events from the neural perspective and the contraction events, the sliding filament model, in the muscle events. So this is a real rough view of skeletal muscle contraction, figure 12.7. You want to go into detail, and of course I have loaded 3-part video that you also have available on Mastering, the A&P flicks. What you're seeing here is also illustrated in figure 12.10 in so many ways. There's certain pictures I prefer to use, so this is basically showing you a neuromuscular junction. Here's the synaptic knob at the axon terminal of a somatic motor neuron, so we have a close-up. This synaptic knob is forming a synapse with the sarcolemma of a skeletal muscle. We call this combination or this communication site a synapse. Specifically, our synaptic knob is communicating at what we call the motor end plate. The motor end plate is a specific location on the sarcolemma where we find a lot of ACH gated, chemical gated channels. Now, the motor neuron has an action potential traveling down its axon and then ultimately causes the opening of voltage-gated channels on the neuron, in fact, on the synaptic knob. Calcium enters the synaptic knob and causes, via exocytosis, the release of our neurotransmitter, which for skeletal muscle is always acetylcholine. Acetylcholine is released into synaptic cleft at the motor end plate. Acetylcholine binds to the acetylcholine receptors on the sarcolemma at the motor end plate, which opens these chemical gated channels. The opening of these chemical gated channels allows the greater influx of sodium than efflux of potassium, and we have a depolarization occurring at the motor end plate. So we need to check to see what happens right after the motor end plate, and you can continue with your figure 12.10. So we have the depolarization happening due to a chemical gated channels opening, and sodium efflux is greater than potassium. Sodium influx is greater than potassium efflux. And what happens is it depolarizes this patch, and the patch depolarization causes the opening of the voltage-gated channels and the adjacent patch to the motor end plate. The voltage-gated sodium channels opening allows a greater influx of sodium, and so this patch depolarizes. And that depolarization causes the next patch of voltage-gated sodium channels to open. And it's in that way that our depolarization wave occurs and travels down the sarcolemma, down the T-tubule, and then at the triad, at the cisternal termia, this action potential depolarization will cause the opening of very special calcium channels. These voltage-gated calcium channels are located within the sarcoplasmic reticulum deep inside the muscle fiber. And once these channels are opened, the calcium stored in the SR will be released to then participate in the sliding filament model by binding to the troponin complex. And that is something that we'll go through again, of course. This is just a very brief overview. You also know that once acetylcholine is released, shortly thereafter, the enzyme acetylcholine esterase will take it away, which initiates the repolarization of the motor end plate, and the opening of voltage-gated channels has finally occurred. And following this depolarization wave of positive charge, due to the opening of now the potassium channel, will have a repolarization wave following. And it closes all the channels, and it will close the calcium channels, and then that means there's no calcium available, which will be sequestered back to the SR to allow the sliding filament model to occur, and the contraction is over, right? That's a lot of information. Most of this has occurred in the nervous system in terms of the physics of it. What is an action potential? A transient depolarization event that includes polarity reversal of the sarcolemma, and the propagation of an action potential, an electrical current, along that membrane. In terms of our muscle, it'll be the sarcolemma. It is an electrical current. It's a signal. It's the means by which the nervous system communicates with the muscles. And an action potential is only generated by neurons and muscle cells. Now, as a reminder, you have this figure here, which of course is in the in chapter 8. And you see an action potential and how the action potential occurs in terms of the events or the phases. We have sodium channels that are opening. Initially, chemically gated channels open, sodium influx is greater. It depolarizes the sarcolemma to threshold, which is usually around -55. And at that point, we have our sodium voltage gated channels opening. More sodium enters the muscle fiber. We call that the depolarization phase. The sodium channels will close once acetylcholine esterase has been removing acetylcholine at the motor end plate. That causes the closing of the sodium channels. First the chemical, then the voltage-gated channels. But at this point, our slow opening voltage-gated channels, which were stimulated to open at the exact same time as our sodium channels were opening, but they're so slow that they're finally opening up by the time the sodium channels, voltage-gated sodium channels, are closed. So now our voltage-gated potassium channels are open, and of course the concentration inside the cell of potassium is higher, so potassium will leave. This is what explains the repolarization event. And because these voltage-gated channels open very slowly, they're also closed very slowly, and by the time they're completely closed, the muscle is in a state of hyperpolarization and needs the sodium-potassium pump to bring the muscle cell back to resting membrane potential. And an action potential has occurred, and you've been able to explain that. So if you're taking a look at 12.1 and you're seeing this depolarization occurring at the motor end plate due to chemically gated ion channels opening or ligand opening. So this depolarization, right, your sodium ions are so positive that they cause the positivity to occur in the next patch of the sarcolemma, and that'll open my sodium channels, and sodium will come in, and it'll also trigger the opening of the potassium, but it just takes a very long time for that to happen. And so you first have the depolarization wave propagating down the T-tubules, followed by the repolarization wave propagating down the T-tubules. Ultimately, opening and closing of channels is the events. And remember, the only reason this is happening is so that we ultimately get to the T-tubules right here, where we're opening, this is the reason, where we're opening the calcium channels that allow calcium to leave. And once calcium is left, we can look at, okay, what happens next? Let's do a little checkpoint. Describe the neuromuscular junction. You can do it just like I did. You can do it slower. You can take it with bigger explanations, but you have to describe the neuromuscular junctions by using proper terminology. I want to hear synapse. I want to hear synaptic cleft. I want to hear chemically gated channels, acetylcholine receptors, acetylcholine esterase. I want to hear motor end plate. I want to hear those terms. How would a drug that blocks acetylcholine release affect a muscle contraction? How would a drug that blocks ACH affect contraction? Think about this. Write it down. Bring it into Zoom. Number 9. What would you expect to happen to a resting skeletal muscle if the sarcolemma suddenly became very permeable to calcium ions? All of a sudden, for no particular reason, calcium is entering the cell. What would you expect to happen here? All right, so give that some thought. Now, let's go to the sliding filament model. This is a figure that you can go along with. 12.9 is similar, not exactly like this, but very similar. You basically have an illustration that shows you what can happen when calcium has been released. When calcium has been released from the SR, it binds to the troponine complex within the thin filament. That troponine complex pushes tropomyosin slightly to the side, kind of pulls on it, and exposes what is called the binding site on the G-actin protein that had been blocked in the absence of calcium. So now that it's exposed, the myosin heads of the thick filament have a great affinity to bind to that, and that's exactly what they do. And that's what you're seeing in this first stage. So at that stage, we have the myosin head attached to the G protein of the thin filament. That's because the calcium block has been removed. And we call this combination cross-bridge formation. So we're trying to understand, how does a muscle contract, right? We know we need muscle, we know we need calcium, and we know we need ATP. So right now, we need ATP to get the head to swivel. It's cocked into place, and it now has to pivot and swivel. And for that, we need ATP to bind to myosin, and myosin will then hydrolyze it into ATP, ATP into ADP plus inorganic phosphate. And that really strengthens the cross-bridge formation, meaning the connection between the myosin head and the actin, okay? Now that they're nicely tightly connected, we do need some ATP to allow that power stroke, right, that swiveling to occur. And that's exactly what happens in this position. So the swiveling occurs. That's great. And now we have that sliding of thin and filament occurring. And up here you see the sarcomere. And during that sliding, the H zone decreases in the space between. And that allows you to understand that the muscle is shortening. Now, ATP is necessary for the detachment of the myosin head. And so we need some more ATP so the myosin heads can detach. So no more cross-bridge formation. So it's slid, it detaches, and it attaches at another location again. Again, we would repeat this cycle with the need of ATP. The myosin head swivel, and that's the power stroke. They release ADP, because they've used up ATP, so they release ADP, and that allows it to detach. So we need to understand that we need ATP and calcium presence for the sliding filament model to occur. And to get you to think about this, I want you to think about what in some cases this is referred to as rigor, when this is the first binding occurs. No ADP, no ATP, we just have ADP. Myosin heads are attached to rigor because rigor means stiff. But I don't want you to think about it that way. But what I do want you to give it some thought is, what's rigor mortis? Rigor after death. The body is not alive anymore, it's dead. How is it that muscles are still contracting? I want you to give that some thought too. So this is the sliding filament model, right? So between thin and thick filaments. And here's a quick excitation-contraction coupling overview. So the events from neuron stimulation to muscle contraction is what the excitation-contraction coupling is all about, the entire event. Now, the action potential is generated at the motor end plate of a muscle due to the stimulation of a somatic motor neuron. The action potential propagates or travels along the, I'm sorry, this should really say, hold on. So the action potential travels along the sarcolemma. Please change that on your outline. It said sarcomere and it should say sarcolemma. So the action potential travels, propagates along the sarcolemma to the T-tubules and down the T-tubules. There, the action potential will open the voltage-gated calcium associated with the sarcoplasmic reticulum, which stores calcium, and now this calcium can be released. The calcium will bind to the troponin complex. The blocking action of the tropomyosin will be released due to calcium binding, and our sliding filament model will occur. Contraction via cross-bridge formation, ATP hydrolysis, and the filaments slide across each other within each sarcomere unit, which repeats itself throughout the fiber. It's this part that we call contraction. The removal of the calcium is by active transport and will take the calcium and store it back in the sarcoplasmic reticulum. It will recreate the troponin conformation change and allow the tropomyosin blockage to be restored. The myosin heads cannot bind to actin, and your contraction ends. The muscle begins to relax. I will let you digest this and post part 2 right after. So this is a lot of the physiology. And before I go, I will give you my little lecture crumb for this lecture. And the crumb will be, I am having broccoli and steak for dinner because it's Sunday and I just feel like having a steak. All right? Bye.